Hello, my name is Dr. Oliver Hardick and I'm the CEO of Puridity. Today I'm going to introduce the FibroSelect Protein A chromatography technology, which we've developed over the last 4 years. Puridity is transforming purification productivity whilst making minimal change to current practices. Understanding the need for the bioprocessing industry to make step change improvements to meet the evolving healthcare needs, while recognizing the established conservative nature built around product safety and product efficacy. This has allowed us to develop the FibroSelect technology so that it's industrially acceptable and ready to implement into bioprocesses. Puridity is based at the Stevenage Bioscience Catalyst, which is about 20 minutes north of London, and we have a team of around 20 scientists operating across 3 different laboratories. Protein A chromatography has played its part in the growth of monoclonal antibody therapeutics. It's such a robust unit operation in terms of providing a highly pure product, irrespective of the feed, that it is used as a platform process in nearly all mAb manufacturing cases. Yet it's something that has remained heavily discussed. This is because despite doing a very good job, which makes it worth its cost, that upfront cost can be considered similar to a capital expenditure that needs to be amortized over many, many batches, for example, over a year of processing. The issues arise when the user is not able to get the full lifetime out of the resin, and this has obvious disadvantages for early stage bioprocesses like clinical manufacturing. But perhaps more importantly, the slow process limits the manufacturing flexibility and fundamentally makes end-to-end single-use bioprocessing unattainable. So in our eyes, there's nothing wrong with the protein A ligands themselves, but it's what the protein A is immobilized to. FibroSelect is a platform adsorbent technology that we've developed from cellulosic nanofibers. The solid 3D structure created has a highly accessible large surface area, which results in a scalable material that performs with the dynamic binding capacities of leading bead resins, but at much higher flow rates. The macroporous structure is also less prone to fouling and suitable for macromolecular targets. This slide goes into a little bit more detail around the underlying material, but in the interest of time, I'll largely skip over this and leave you to come back to me with any questions. Importantly, the functional material formed has a good homogeneity and provides intrinsic flow distribution. The porosity of the FibroSelect material is comparable to a packed bed resin at around about 70%. These properties enable the exact same protein A capture process to happen in seconds rather than minutes, which means we can complete a full chromatogram cycle in 1 to 4 minutes, depending on the system that we're operating the unit on. This is down from the hours that it usually takes in an industrial process. This speed enables the end user to operate in a rapid cycling mode, which means that the lifetime of the protein A resin, let's say 200 cycles, can be exhausted over a single shift. This means that the end user can either reduce the size of the unit operation 100-fold, as we've done in the case of this example in this slide, and then discard the FibroSelect unit in a single-use manner, or the end user can complete the unit operation in a fraction of the time. It's the flexibility of processing which is the real benefit here, with the end user being able to specify how they want to run their operation to best align with their other unit operations and processing needs, rather than being stuck with this paradigm that chromatography takes a number of hours. Typical columns have a productivity of 10 grams of monoclonal antibody purified per litre adsorbent per hour. In a multi-column system, that productivity of packed beds can be improved from anything to 20 to 60 grams per litre per hour. FibroSelect has a productivity of many hundreds of grams per litre per hour operating on a standard single-column chromatography rig, and this is down to the high throughput and high dynamic binding capacity that the FibroSelect material exhibits. With the same inputs in terms of feed streams and the same infrastructure in terms of chromatography rigs, FibroSelect achieves the same outputs in terms of the critical quality attributes of the product being purified. This, for the first time, enables single-use product capture applications using proven methods and materials, and this has been a major driver for our extensive interaction with leading biopharm manufacturers. Here we see the dynamic binding capacity plotted against flow rate. The dashed lines are commercially available 1 milliliter pre-packed bead resin columns. As you increase the flow rate, you see an expected decrease in dynamic binding capacity due to the flow and diffusional limitations of these adsorbents. The solid lines above represent FibroSelect material coupled with the same ligands as the resins below. Here you can see good dynamic binding capacity is achievable, and critically, the maintenance of that DBC with increased flow rate. This next slide demonstrates where we operate FibroSelect in terms of the flow rate, and you simply can't operate a packed bed at this speed due to the pressure limitations of current columns and resins. So what happens when we operate this rapid cycling fashion with true industrial feed streams? Here we have two examples of different monoclonal antibodies that we've purified overnight with FibroSelect at an industrial end user's site. In both cases, the load challenge exceeded 40 grams per litre, and after 200 cycles, the performance of the unit was maintained above 99% in terms of the recovery. In both cases, no clean and place steps were used, and the feed was at a late stage development and well defined, which meant that it was quite a clean product. With just a 0.25 milliliter Protein A FibroSelect unit, we were able to purify over 3 grams of monoclonal antibody in this time. With that, we had equivalent purity and yield to the packed bed control column. You can see in this case we're approaching the kilogram per litre per hour level in terms of productivity. We're working with 12 industrial partners today, including a number of top 10 players, and we typically see the same strong lifetime performance over 200 cycles without requiring CIP. But there are examples where feed streams are less defined and do cause fouling issues, in which case we employ 0.1 molar sodium hydroxide CIP to manage this. Our focus has been to rapidly innovate and iterate our technology with the continued support and input from industrial end users over the last 3 years. To date, we've been working with end users up to the 100-litre bioreactor scale, and we expect to be able to support purifications at the 500-litre bioreactor scale by the end of 2017. In 2018, we'll be looking to support a 2,000-litre sub-unit in GMP processes. That means this technology would be ready for the market. As a platform technology, our end users have driven complementary development into different routes of operation. This is an example of a salt-tolerant anion exchange flow-through polishing unit that we've operated to high loading challenges. Unlike traditional AEX membrane adsorbers, there's also an additional opportunity here of operating in the same rapid cycling fashion to maximize the product quality and still get the high productivity from the unit that's being operated. We've also developed high throughput tools that can be used by those groups who favor working with robotic systems to generate process development understanding. Here's an example of our 8-well module unit that can be used on the fixed tip Tecan liquid handling system and operated in a mini column format. An example of this is given here, where a Protein A post-load wash study was carried out screening 8 different wash buffer conditions in just 10 minutes. We were also able to generate a faux chromatogram by dropwise collection of the elution peaks. However, in terms of process development, most of our collaborators prefer using the research scale syringe filter units, which are also able to generate a huge amount of data. In this case, we carried out a full factorial design of experiment study for a Protein A elution optimization, performing 51 ACTA runs in just 3 hours. Obviously, this capability requires careful consideration of subsequent offline analytics. As a final example, here we worked with GSK to carry out a fully integrated high-throughput platform process. 48 15mm amber bioreactors were centrifuged in situ and then placed onto the Tecan deck, where the supernatant was collected and delivered into the FibroSelect microunits under positive pressure. The purification was successful without any pre-clarification and yielded enough material for the subsequent analysis of the material generated. So in summary, FibroSelect enables effective single-use product capture purification for the first time through rapid cycling operations. This requires the required inputs and performance outputs in terms of product quality rely on the well-characterized chemistries like Protein A, and thus remain in line with what manufacturers expect to see from current chromatography columns. The flexibility offered to manufacturers thinking of redesigning existing processes or implementing new ones enables entirely new processing strategies to be considered that meet the needs of both the industry today and thinking ahead in the future. Direct feedback from end users is driving FibroSelect Protein A forward as our first product to market by combining the well-understood purification advantages of single-use technologies and unmatched productivity levels of FibroSelect, enabling flexible production scenarios. The performance of FibroSelect facilitates a wide range of differing applications with opportunities beyond what we've achieved today. that, all I have to do is thank the people involved generating the data and supporting the development, both from Puridity and GSK, and to thank you for taking the time to listen to this webinar. I'd be happy to answer any questions that you may have, so please do get in touch.